Hello everyone, my name is Jake Piland, and today I'll be answering the question, does dynamic network analysis improve protein 3D structural classification? Here's the outline of what I'm going over today. First, I'm going to define our task, protein structural classification, or PSC, and cover related work. In PSC, sequence or 3D structural features of proteins, along with predefined structural classes, are used to train a classification model to learn which features correspond to which classes. This model can then be used to predict structural classes of currently unclassified proteins based on their features. Despite existing technology, the functions of many proteins remain unknown, so we need to computationally predict them. Luckily, protein structure correlates to function. So the big idea is that through PSC, one can correctly analyze the features and identify the structural class of a protein, which then correlates to function. Note that we do not proceed with predicting functions in this study. We stop with evaluating if we can correctly predict structural classes. Traditional PSC relied heavily on sequence-based features. But because two proteins can have different structures despite having similar sequence, and vice versa, 3D structural features were later included. We recently proposed a series of approaches that model proteins as protein structure networks, or PSNs, for the task of PSC. A PSN represents a protein by representing its amino acids as nodes and connecting those nodes that are sufficiently close in the protein fold. This allows us to leverage powerful graph-based features for classification. Now, there are different characteristics that a PSN can have that I need to define here before being able to explain our contribution to PSN-based protein structural classification. The first is unordered versus ordered. An unordered PSN, like the toy example shown here, has unannotated nodes. Even though they correspond to an ordered amino acid chain, we cannot see that order. Whereas an ordered PSN has nodes numbered according to the position of their corresponding amino acids in the amino acid chain. Next, there is unweighted versus weighted. Here we'll focus on the edges. An unweighted PSN, like the example shown here, has edges without a weight value. A weighted PSN has edges with weight values. For each edge, its value, or weight, is a function of sequence and 3D distance, such that the largest values are assigned to those amino acids that are both far away from each other in the amino acid chain and are spatially close to each other in the 3D fold. This way, the heaviest weights go to those nodes that are close, but not just from being next to each other in the sequence. This kind of relationship is important as it can indicate a fold. Our first PSN-based PSC approach used unweighted, unordered PSNs like you see here. We will refer to these as original PSNs in the paper, and I will do so here. We also tested ordered, unweighted PSNs, and we refer to these as ordered PSNs. These were defined in these two studies, although the first one deals with a task other than PSC. Then there was unordered weighted PSNs, which we refer to as weighted PSNs. Note that there is currently no ordered weighted PSNs for the task of PSC or at all. These are the three PSN types that we will compare against in this analysis. And the reason we only compare against PSN-based approaches is because in these two studies, we did compare to traditional sequence and 3D structural approaches. Specifically, for sequence, DAA composition, AA composition, and SVM fold, and for 3D structural, TM align, Dolly light, GIT, and GR align. And in these two studies, PSN-based approaches were established as a state of the art. Now, we have these great PSN types, but they are all static PSNs, which capture the final native structure of a protein. However, protein folding is not a static, but a temporal process. So how can we do even better at capturing proteins? By modeling them as dynamic rather than static PSNs. In order to model PSNs as dynamic, we need a new PSN type, which I will describe in this section, namely dynamic PSNs. I will define these and show an example of their construction. Again, one attribute the three previous approaches all had in common was that they were static PSN approaches. A static PSN represents a protein's final native structure as one network. But a protein folds as it is built. It has emerging substructure. We aim to approximate a protein's substructural configuration using a dynamic PSN, with the hypothesis that a new approach that uses dynamic information will improve on static PSN-based PSC approaches. Intuitively, a dynamic PSN is made of snapshots. The first snapshot captures the initial portion of the protein, and the next snapshot captures a larger portion of the protein than the previous snapshot. This continues until the last one, which captures the full native structure. Let's go into detail with an example construction. We tried two different techniques to construct dynamic PSNs. While we talk about both in the paper, one performed much better than the other, and it is the one that I talk about here. Here I have an amino acid sequence. For reference, here is the final static original PSN of this toy protein. The color gradient corresponds to the position in the amino acid chain, with white being the beginning or end terminus of the chain, and then darkening to black for the end or C terminus of the chain. One of the parameters for this method of construction is the number of amino acids to add per snapshot. For this example, I'll add 2 amino acids at a time to build each snapshot. In the paper, we add 5 amino acids because alpha turns and beta strands are roughly 3 to 7 amino acids long. Anyway, here's the first snapshot of our example. And then the second. Here is the third snapshot. And the fourth and final. As promised, the full native structure is captured. To be clear, this whole collection of snapshots from left to right is one dynamic PSN. Now, let's put our new PSN type into perspective with what we covered earlier. Remember this table of PSN characteristics? Recall that all the previous approaches dealt with static PSNs. Now I will add the row that contains our new type. Our dynamic PSNs are unordered. They are unweighted. And by definition, they are dynamic. We refer to these as dynamic PSNs. Know that the reason we propose dynamic PSNs as unordered and unweighted, as opposed to ordered or weighted, is because we have methods to analyze unordered, unweighted dynamic PSNs. Availability of methods to analyze ordered dynamic or weighted dynamic networks is limited, if not entirely unavailable. So we use this PSN type to test our hypothesis. And if dynamic PSNs perform well under unordered and unweighted constraints, we can hypothesize that they will perform even better as ordered or weighted dynamic PSNs. But that is the subject of our future work. I will add colors to show where the other approaches differ from dynamic PSNs. Green shows the same characteristic as dynamic PSNs, and red a different characteristic. Note that original only differs from dynamic in one characteristic, which is the key one. Thus, we need only compare dynamic and original to test our hypothesis that adding dynamic information is useful. However, we will compare to the current state of the art, ordered and weighted, to further test the potential usefulness of dynamic PSNs. All right, I have covered our new PSN type. Next, I will describe the methodology to test if dynamic PSNs improve on static PSNs. The hypothesis we test is adding dynamic information to PSNs improves on PSC performance compared to static PSNs. For the data, we extract protein structural data from PDB. We use this to construct a PSN for a protein domain. We get domain structural labels from the Cath and SCOPy databases. These databases have hierarchies that feature increasingly detailed classes. I'll explain using a generalized example that could fit either database. At the topmost level is the most general labels, such as mostly alpha, mostly beta, a mix, and so on. Then in the next level, for each label in level 1, there's a set of more specific labels. So now we have an alpha 0.1, an alpha 0.2, a beta 0.1, a beta 0.2, and so on. This continues. In level 3, there are even more specific labels for each of the labels in level 2. And finally, the most specific labels are in level 4. Now we could make a data set out of any one of these green boxes. In other words, for each of these boxes, we have a PSN data set. We could test and train on all of the beta proteins just as easily as we could on the alpha 0.1.2 proteins. Now, some of these green boxes don't have enough data or are too specific. We filter these out, which leaves us with 71 PSN datasets from all 4 levels of Cath and SCOPy. That is 71 opportunities to test and compare the performance of dynamic and static PSNs. Because we're dealing with networks, we are extracting features that have been prominently and successfully used in a variety of computational and applied tasks, namely graphlets, subgraphs of a network. The advantage being that we have graphlet-based methods for each of our PSN types, which offers consistency, meaning that differences we see in performance are due to differences in the PSN type and not feature type. For our classification, we use 5-fold cross-validation. We split a given PSN dataset into 5 folds, or subsets, maintaining class balance. For each subset I, we train on the union of the other 4 subsets. We optimize the hyperparameters of the model, and we use the trained model to attempt to predict the structural classes of the PSNs in subset I. We consider a PSN from the test data to be correctly classified if its predicted label matches the actual label and incorrectly classified otherwise. We use a logistic regression classifier. As we achieve high performance with just this simple classifier, we do not currently investigate using more complex classifiers. The performance measure used is misclassification rate, which is, for each dataset, the number of PSNs classified incorrectly divided by the total number of PSNs in the dataset. Note that that means a lower misclassification rate indicates a better performance. That covers the details of the methodology and evaluation. It's time to see the results. Here on this graph, the y-axis is misclassification rate. Remember that a lower value indicates a better performance. The x-axis is our 71 PSN datasets. The first one's being Cath, and then the next one's being SCOP. The vertical dashed lines indicate changes in hierarchy. We have Cath 1, Cath 2, Cath 3, and Cath 4. We also have SCOP 1, SCOP 2, SCOP 3, and SCOP 4. The colored lines indicate the different PSN types, with red being dynamic PSNs, blue being original, gray being weighted, and black being ordered. Just looking at this graph, we can see that dynamic outperforms the other 3 on a number of PSN datasets. Specifically, dynamic outperforms original on 63 of 71 datasets and ties on 4. It outperforms weighted on 61 of 71 datasets and ties on 2. And dynamic outperforms ordered on 51 of 71 datasets and ties on 13. This means that the dynamic is only inferior on 4, 8, and 7 datasets respectively. And this is statistically significantly better performance. A simple set of results to indicate a powerful conclusion. Yes, adding dynamic information to PSNs improves on PSC performance compared to static PSNs. This opens up opportunities to explore protein folding related research questions using dynamic PSNs. Finally, I would like to acknowledge and express sincere gratitude for the support of my advisor, Professor Tiana Malakvich, my coworkers at the Complex Network Lab at the University of Notre Dame, and our collaborators, Professor Patricia Clark, Professor Scott Emmerich, and Professor June Lee, and our funding from the NIH. Here's our archive reference, and thank you very much for listening today. Are there any questions?